Man, I watched that video and I just get tingly in a good way because it really was just this amazing, fantastic experience. I wanted to address the one question, and maybe we can talk a little bit about it because this was a little bit of a struggle. In terms of I can't find it now. It was about reagents, about the students designing their own experiments and then needing to order the reagents. And so the way I set up and scheduled my class was that there was one day where they finalized their standard operating procedures, and then they actually had to go online and they had to get me what I called a grocery list, a shopping list. Okay, so here's the question. Something I am struggling with is the openness of having the students design the experiments and then having the reagents available to test the question. What has your experience been with this? And so I actually had the students with their student after they designed their experimental design and we went through it and designed their standard operating procedures, then they spent some time with the shopping list and what do they need, what do they want. And we had a very accommodating and helpful team that immediately turned around and was able to order the reagents and get them to us quickly. This was pre-COVID times. I know things are back ordered for 17,000 years at this point. And so if I had to do it again, I might try to constrain it a little bit more so that we have a very well stocked teaching supply area. And so try as much as possible to engage that because I mean, my favorite yellow tip is back ordered until November right now, which you probably think I'm a weirdo because I have a favorite yellow tip, but that's what it is. So that was how I dealt with that. Ken, how did you deal with the students wanting to order this, order that, or how did that work?
So the way I tried to deal with that is in the setup of the course itself. I have students, bring their proposals to the class for a peer review. But before that peer review, the students then, like what you did, is I have them give me a shopping list, and we figure out what's reasonable about it, and I go over it with them. Obviously, some things we're not allowed to have. Even though my students did research on a uranium mine, they were not allowed to buy control uranium. That's how it is. But generally, it's working with the timing. It's working with your whoever does the support for your labs. And I think we're in a very fortunate age, at least before the plague, to have the kinds of overnight delivery services from those huge mega companies that we have that make this stuff a lot easier.
So I took a little bit of a different approach to this in that I had a certain amount of equipment ordered, sampling equipment, things to measure nectar. And so I let the students know what was available prior to them developing their experiments, because a lot of the equipment they would need is a little bit more expensive and takes longer to order. So I kind of constrained what they could do based on what I had available. So that's it.
And then I did a combination of what Dr. Marshall, Dr. Foltz-Sweat, and Dr. Sweat did, because I had students who, of course, wanted to run DNA, and getting a genetic analyzer like that is kind of hard, and of course it's going to be in the research space. And so again, shopping list, but also let them know ahead of time, some of these bigger cosmic questions like can we get measurable DNA from a fingerprint may not be able to happen at this point in this iCure.
So for my 4-week mini cure, I still wanted to give students options of what they could test, but we didn't have time to wait for deliveries. So I made sure to work with the folks in the teaching labs and use reagents that they had on hand. But I also encouraged students to look around for what was available locally. So what could we get from the local grocery store? What could we sample out from the environment to be able to get quick access to whatever it was that they want to study. For the larger Drosophila development and behavior cure that I did this past semester, I did a lot of what my colleagues did. I did have one kind of weird case where I had some students want to test fly behavior, but they needed this kind of elaborate contraption that was way too expensive for what we were trying to do in our course. So I encouraged them to think about how else they might be able to test it. And they actually took it upon themselves to go out to the local hardware store, find the materials that they need, and let me know this is what we need and this is how we want to assemble it. So I encouraged them to be creative about how they can solve these issues if we can't order what they need.
That's great. Yeah, I think that each one of us had slightly different ways to approach this, but I think that that really worked. And the reagents that I was ordering, at the time were typical things that Fisher had on hand, and our Fisher orders are really pretty quick. And I know I don't own any Fisher stock. So we have a great question here, which is what prep do we need to do on how to deal with failures?
I talk about it ahead of time, and I preface it that in forensic science, there's information and you don't know the answer, that there are only a couple of parties that know the answer. One of them's not talking for one reason, and the other one's not talking for another reason. So I just bring it out up front that you may not get results. That's okay. That's the nature of science. That's the nature of doing these experiments. And it's not necessarily failure because you do have results. You do learn something from it. So I try to put a more positive spin on it while also kind of giving them a heads up at the beginning.
So what I've done to address that, and of course this takes a while until you've had a few cures, but I have a large database of bee data. So like Kim said, I talk to them about the fact that it's not necessarily a failure, and I have them evaluate their experimental design or their methods that they use. But in terms of data analysis, I can have them access this larger database that I have and ask a question that my students haven't necessarily asked before. And that will still give them data to do the analysis part.
So there's two strategies I took to tackle this issue. First off was pretty early on in the semester. As they were developing their protocols, I kept asking them, well, what if this doesn't work? And what if this doesn't work? So that they could start thinking about what the next steps would be. Because that's something we have to do in the lab all the time. If this experiment shows something, then okay, we can go off and tackle that data. But if it doesn't work, then what are the next steps? What other types of things could we try? So I play the what if game a lot with my students early on. And towards the end of the semester, if things didn't work, I asked them to focus on what they did learn. And a lot of times that just goes back to really emphasizing the scientific method and understanding the process, and also understanding the fact that negative results, as my colleagues were saying, negative results are results. So they still learned a lot, even if they didn't get the results they were looking for.
So I agree with everything my colleagues do because I use a lot of those strategies. one of the other ones that I have come across, because I've been this long in teaching, that I find to be very effective, is to salt the class with stories of your own failures, right? I tell the students I'm very proud of my work in organic chemistry because I consistently had about 110% yields on all my experiments, right? I talk to them about other things, and to be honest, I will sometimes take the mistakes my students have made, and as I tell the story to the next generation of students, they become my mistakes because I want them to see that I am making mistakes and it's not a bad thing. And so I think that kind of background strategy of humanizing it really it helps the students not only accept it, but it also helps them relate better to the instructor.
I think for me, the biggest thing with the students and the failure was that they were, the experiment didn't work quite right, or, there weren't significant results between the two variables. And I had to kind of remind them, you see what the grading scheme is. You've seen all the rubrics. Was there anything in any of the rubrics of the grading scheme that said that your grade was going to be dinged if your results weren't significant? And so I think for my students, a lot of their fear of failure was tied up in their worrying about their grade. And when we just reminded them, this is the syllabus, this is how we're grading, here are the rubrics for each activity, and you're earning an A, and so you will continue to, as long as you continue to, perform the activities, that sort of alleviate a lot of those concerns.
Yeah.
So there was a really good comment in here to go back to the shopping list, which was, we design experiments based on previous work in our cure, so reagents are generally available. And I think that's a really good that's a really good strategy too, especially since there seems to be a lot of things that have been back ordered. It's another really interesting question. Has anyone had to deal with obtaining ethical approval before student studies could be conducted? And so what tips do you have for accomplishing that in a timely manner? And so Kim wants to chime in on this.
Yes, so my students wanted to use bodies to see how long saliva lasted on buried bodies. So thankfully, Dr. Marshall was also working during the summer. So I'm asking her, we can't get dead bodies. What do you suggest? So we were looking at the frozen rats. And this was all happening within a couple of days. And so finding out what the students wanted to do ahead of time was very helpful, because then I could start calling and asking people, because I did need to contact IRB, and instead of just the Institutional Review Board. So instead of just relying on emails back and forth, I am getting people's numbers and I'm calling them. And I had all of that information ahead of time. And again, thank you to Dr. Marshall for that. How were these animals euthanized? What are they being used for? And because even, I don't know if this is an urban myth or not, but I guess even if you go get chicken or beef from the butcher, that could be an issue. So I had this question and I got an answer in, honestly, a fairly short time. I'd say about 2-3 days. I had an answer that yes, I could use these particular specimens. You don't need IRB approval. And when I had talked to the representative from IRB, they said, just if we have to get this a quick turnaround, just make sure all your I's are dotted, all your T's are crossed, complete SOPs, and we'll try to, expedite the process.
I just wanted to quickly add, I appreciate you asking that question, because we should be integrating a lot of ethical concerns throughout any of the cures, because even you know, sometimes I think we think of, you know, the dead bodies needing some other protocol. I mean, you can tell in the statistician that doesn't even know what half of these reagents are. So at any rate, but there's also ethical concerns and like social science questions. And you can't just go out and ask your classmates things at the university because that's just not okay. Like, so certain things that they think, I mean, students might think that, you know, they might have to ask permission to get a dead body. They also need to ask permission for a lot of things. And so, and there's so many, I mean, I work with so many socials. I mean, and a lot of you guys will do social science type experiments, and the ethical concerns about those are different but equally valid. And so, and also require, we can't ask people about their grades, we can't ask people about their socioeconomic status, we can't ask, I mean, we can't just send out a survey to the classroom. So they need to know that. They need to know how we can ethically and, you know, like, and follow the rules, right? Like, and follow the guidelines that are set forth. So I think sometimes it's super obvious in Kim's situation, but not so much when they think they can just ask all their friends, because then we are at the university and we are bound, we have to protect our students.
So if I could add to that, yes, it's very important you have these kind of ethical, these ideas in your cure. Ethics in science is really important. And it's important students understand that as well. All of the things that Jennifer Broach, Dr. Broach was just talking about in terms of what information we can get from students are very important for them to understand and realize. My advice to you is, if you foresee this happening, if you foresee that you would need institutional approval for a cure, is to start working on it as soon as you can. And to start, you know, even before the semester you're teaching it, start building those relationships with your institution's IRB. And also, you know, what I would recommend for like the social scientists that are here is a mini cure that's focused on the IRB process, right? So yes, you have a research question, but you could actually use that as a mini cure in a social class to teach students about the IRB process, about how you can ethically harvest data from human beings. But as a final note, this is also why I work with plants.
So I did want to follow up. We, when we were awarded the grant from the National Science Foundation, before we were able to activate the grant, we had to have our IRBs in place. And so we do have IRBs in place because we are asking and answering questions about students' trajectories, students' careers, and students' ability to complete a degree with and without the iCure, as well as some of their demographics. So we do have that in place. ASU has a very robust infrastructure for animal care and use, human subjects, you know, IRB, as well as recombinant DNA. And so we have many websites and many trainings that we have to take in order to really be able to, you know, do the experimentation that we need to do. And I see something in the chat that says, I definitely see the merit in working on the ethical approval for fall now. And I wanted to share, not with the iCure, but I had an honor student who wanted to do her thesis. And I walked her through it, and she wanted human saliva samples to do some microbiome. And I told her, sure, but you have to take city approval. You have to write the protocols. You have to write the IRB. And I will submit the IRB. And she was really taken aback. And it was a lot of work for her, and I helped her. I didn't, you know, send her adrift. And then at the end of it all, I said, you know, I did that on purpose. It was very difficult, but now you have something that you can use for your medical school application that sets you apart and above and beyond everyone else. And so I think at the time it was all happening, there were words that she was saying that she didn't want me to hear. But at the end of it, she actually thanked me, you know. So sometimes work is hard. Let's see. So we had, we just completed 2 semesters of analyzing the nasal microbiome of students who tested positive and negative for SARS carriage, and we walked students through the IRB process. Yeah, I bet that was a lot of work, and I bet while the students were doing it, it was complicated, but I bet they learned a ton. And I tell the students, I said, you just took a bunch of safety classes. This is for our summer program. Those should go on your resume. Like, oh yeah, that's a good idea. So try to, you know, talk up the positives. You've now done something that people you're competing for haven't done. And we did have something in the chat that said, if you're meeting the student learning objectives, but the students don't have papers, then they're at a disadvantage for jobs. I can see that. But if the students play up what they have learned and what they can do independently, I think that is a real positive as well, because there's a lot of things that I do in my research lab that I haven't gotten papers for. Let's see. So we got another question that actually is a really good lead into our videos that will be coming in a few minutes. It is, does iCure include ecology or environmental questions that do not require many lab supplies?
So I can go ahead and speak to that. So my pollination biology cure is obviously environmental, and it's usually field work. It doesn't require a lot of supplies every semester, but there is an initial investment in equipment. Like, for instance, I have equipment to measure sugar content in nectar. I have the little capillary tubes, nets, pan traps, all of those things, but I can reuse these every semester. But absolutely, you can use iCures to ask environmental or ecology-based questions, and I highly recommend that, being focused on pollinators.
The other thing is James Filikowski, the one that talked initially on the Successes and Challenges video. His research is on sharks, and we did not send all of the students out to the ocean to to go study sharks. So they were all analyzing very unique research questions, but from previously collected data. And that's okay, that's fine. And there's also, so they did a whole cure on previously collected data. They still have to do the same things. They just don't go through the physical steps of collecting the data, but that doesn't make their cure any less scientific, or that whole research, it doesn't change much of the research process. And it could also help you even speed it up from a social science perspective as well. Like, there are a number of databases out there that are national databases, you know, crime victimization surveys, you know, there's lots of really cool questions you can ask. I know one of the cures that we partner with Phoenix College, their mini cure, their cures are embedded in already existing courses. They're not full courses. They partnered with the crime lab and the Phoenix Police Department, and they're looking at crime trends and things like that. So they were given a database of, you know, like a huge database, but they weren't given specific research questions. They knew a methodology that they were able to do, but then they were able to ask different questions of the data. So that's always an option, and that doesn't require any tools, because there are freely available tools for analysis online. So That's one thing that you can do, and that just changes. And that also could be for ecology. I know like the IUCN Red List has all of their data freely available. There's lots of available NOAA. There's lots of freely available websites that we had kind of posted yesterday, a few of them during the breakout session, that you can use to do environmental studies or ecology studies. Anyone else?
So yeah, field studies that require relatively little resources, I think actually are an incredible way to bring cures in. You'll notice in some of my phytoremediation videos, I talked about running transects and doing plant sampling. That can be done with just a transect. And in fact, if you dive into the literature on, I'm gonna go a little bit away from plants here. If you dive into the literature on lichens, which is where I did my dissertation research on, lichens are known to be highly sensitive to air pollution. And all of that research was worked out by research studies of literally transects and measuring populations at distances from pollution sources. So there's an incredible body of research already out there about doing these kinds of field studies that really only require you to be able to quantify the numbers of the organisms you're interested in. So I would highly encourage one of those field courses. And yes, it should be really easy to do so with the minimal support, resources, as long as you couch your questions appropriately.
Okay, great. I have put several different links for several different databases here in the chat. So hopefully that gives some people some ideas. Let's see. I think we answered all the questions in the chat. Are there any last questions? We have a few more minutes until our next section. Any last questions? Oh, we posted that twice. Okay. All right. So I don't see any last questions. So I wanted to let you know that our next section is all about using bees as a study model. And these videos are from Dr. Foltz-Sweat. So I should probably throw in another Shakespeare joke here about two bees or not two bees, right? And interestingly, I never knew that there were any kind of bees besides honeybees. And Ken and I were out looking at some rosemary, and he pointed out to me, hey, look, that rosemary's got a bee on it, and it's not a honeybee. And I looked at it, and I was like, that bee is blue. Bees come in blue? So Jennifer Foltz-Sweat, Dr. Foltz-Sweat, Ken is teaching me bee biology. So you've done something right.